Hi, everybody. Happy quarantine. I hope everybody's staying safe and paying attention and making sure that this spread doesn't get any worse. The CDC has said that for 8 weeks, nobody should be at any gathering of any more than 50 people. I mean, that includes going to movies and malls, and there must be a reason for this. These people are smart. They know what they're talking about. So, we got to move on, though. And the reason we have to move on is that IB exams aren't moving as far as I know yet. And so we need to be on track and on point so that when we get back, we can do our last section and everybody will have enough information for the IB exam. Those of you not taking the IB exam this year, many of you will take HL Bio next year, and this stuff could be on it as well. So let's just do a quick recap of everything that we've talked about. First, we talked about these things called chromosomes, and that chromosomes are a combination of DNA and protein. I remember that those proteins are called histones, and that the DNA is kind of all wrapped around it. That the DNA consists of a bunch of little sections called genes, and most of those genes are codes to make proteins, and those proteins are what give you the traits that make up you. We learned a bunch of information about how we can find characteristics of these genes, how many DNA bases long they are, what the protein is that they code for, and so on and so on and so on and so forth. We also learned that because of the universal genetic code, meaning that every living thing uses A's, T's, G's, and C's in their DNA and U's in their RNA, and they all read these genes 3 bases at a time on their ribosomes to make proteins, that we can actually do a gene transfer, which is when we put genes from one living thing into the genome of another living thing. This uses 2 enzymes, one called a restriction enzyme and one called a ligase to make this all happen. We learned that this gene transfer leads to what we call genetically modified organisms, that there's some pluses and minuses to these things for the economy. That in the case of BT corn, the plus is that you get corn that is resistant to or produces a pesticide so that it is resistant to these moths eating them. However, the first strain that was made was very dangerous to caterpillars because the pollen from the corn would blow into neighboring fields, land on the milkweed, and the monarch butterfly caterpillars would eat it and die as a result. However, later strains didn't produce as much, didn't express that gene as much. And then we stopped talking about GMOs and we went into the actual mechanism that causes the genes to produce proteins. And this happens through a process called transcription, which happens in the nucleus, and translation, which happens out in the ribosome. Changes to the DNA can lead to mutations. Not all mutations are harmful. And then the one that you specifically need to know is the sickle cell mutation. You were supposed to do that one in class, finish it up for homework. We checked it out in class, but make sure that you know exactly what the mutation is, that the GAG gets switched out for a GTG, and the glutamic acid becomes a valine. This is important because glutamic acid has a charge, but valine is hydrophobic. So all of that is on the mutations in hemoglobin worksheet that we did. All right. We learned about stem cells, and that stem cells contain all of the DNA for that living organism, but doesn't express every single gene. And so there's a lot of different kinds of stem cells. Most of them we are in the embryo. There are adult stem cells that aren't as potent. They're only multipotent compared to those embryonic stem cells, which are toti or pluripotent. And this is kind of where we're going to pick back up right here. These stem cells, at the start of your development, kind of go to become every single cell in your body. And how they do that? They do that by dividing. They divide to produce more and more and more cells, and they differentiate. And differentiation is when they express very specific genes that allows them to become that cell that they're going to become. And it's this divide part that we are going to be focusing on right now. There's two ways that your nuclei and your cells divide. The first way is called mitosis. This produces genetically identical nuclei to the parent cell. And this is how you become all of these cells that you are. You started off as one cell, then divided into 2, then to 4, then to 8, 16, 32, 64, and so on and so on. And you do that through the process of mitosis. Your body still uses mitosis all of the time. It does it whenever you've got to repair tissues in your body. And by tissues, I mean like these groups of cells that all work together to do something. Like you have tissues in your skin and tissues in your muscles, and you have stomach lining tissue and so on and so forth. So whenever you have to repair these problems in your body, your cells reproduce by mitosis to create new cells to form in those places. At some point you got to grow. Not just emotionally and mentally, but physically. You physically have to get bigger. And you get new cells from the existing cells by this process called mitosis. Some living things rely on mitosis to be the way that they reproduce, and this is called asexual reproduction. Asexual because it doesn't involve combining genes with another living thing. Some plants do this. There's a lot of little microorganisms that do this and so on. And then finally, we'll go back to when you were an embryo and you had to become all of these very specialized kind of cells. That process is called development. And so development is when you make new cells and these new cells become specialized. So this also includes this differentiation up here. So those are the different times that your cells do mitosis. Now, your cells aren't dividing all of the time. Your cells have a very specific cell cycle. We'll do that over here. Okay. Oh, you know what? I should really mention the other kind of cell division. And this is the second kind of cell division. First, let's mention that. It's called meiosis. This is the formation of your gametes, or your sex cells. And these include your sperm or your eggs. And this is these cells that are produced have half the number of chromosomes as the parent cells. So they are N. Their chromosome number is N, which means that they only have one of each chromosome. And this is important because you want them to combine with a cell that also has one of each. So you have N plus N, which is 2N, and that's what all of these mitosis cells are. So your cells are not in mitosis all the time. I don't know why I started this one right in the middle. That seems really inconvenient. So your cells aren't in mitosis all the time. Your cells aren't always going through this. And so let's talk about kind of like the life cycle of a cell. I don't know where to go here. Okay, we'll just go up here. So let's talk about the cell cycle. So this is like a life cycle of a cell. All right. Everybody ready? Okay, we're going to draw like a pie chart here. Okay. Pie chart. And almost all of the pie chart, I'm going to outline in red here. Almost all of the pie chart is the time where the cell is not dividing. And this whole red part of the pie chart is called interphase. Now this has some much smaller parts to it, but let's just focus on interphase for 1 minute here. Interphase is just sort of like the life of the cell. It's doing a lot of metabolic reactions, which just means that it's doing the reactions that it needs to live and survive. And it's growing. And there are two parts where we specifically say that the cell is actually like growing, and those are called G1 and G2. The G stands for growth, because when you want the cell to divide, the cell needs to get a little bit bigger first. There's also a part of the interphase where the DNA needs to replicate. Replicate is a science name for, or the science word for copying. We'll go into some detail about that actually in the next video. But the DNA has to replicate at some point, and this is called the S part of interphase, or the S phase. S is short for synthesis, because synthesis means to make new things. All right. And let's see here. What else can we add in here? Let's drop these phases in here, okay? All right, so I'm going to put these phases in blue here. And let's say that the first phase is this right here, and we're going to call this G1. So this is a part of interphase. So let's do G1. All right, so the cell grows. It makes more organelles, because when it's going to divide, it needs to be able to send more mitochondria and Golgi bodies and lysosomes and all that kind of stuff over there. Does a lot of transcription and translation as it's making all of the proteins and enzymes that it needs to live and all that kind of thing. Does a lot of cell respiration, because all of these processes require a lot of energy, and that's energy in the form of ATP, remember? Also obtains a lot of nutrients, because it's got to grow, and growing requires supplies and all that kind of stuff. So all this is just about growth. This is also true over here in G2. But we haven't gotten to G2 yet, so let's put G2 in here. G2 is going to be this other third of this area right here. I'm going to, hopefully nobody finished that up just yet. G2 is going to have to be a little bit bigger than that, because what we don't need is this other middle phase to be so gigantic. Okay, and this middle phase is going to be S phase. S, remember, means synthesis. And in the synthesis, this is when the DNA replicates. This is really important, because if you want the cell to double, if you want the cell to double in size, if you want the cell to double and create an exact copy of things, like this is incredibly important. And so I want you to think about it like this. So you got this cell that's 2N. Now, if this cell just divides in half right now, you're going to get a cell that's N and a cell that is N. But are these new cells that we just made right here, are these genetically identical to the parent cell? The answer is no, because in order to be genetically identical, you have to have the exact same number and types of chromosomes. And if you just have half of them, there's no guarantee like what that combination of that half is. And so it's not even like a full maternal or a full paternal parent cell. So this can't work. You can't have it like this. So what has to happen is that the cell has to be 2N, and then after S phase, it's going to become 4N. So it's going to have 4 of each chromosome in there, 4 chromosome 1s and 4 chromosome 2s, and we'll go into that detail a little bit later. And then what happens is that 4N cell divides, and now you have 2N cells. And these 2 cells that were just formed are exactly the same as this parent cell right here. And that's the point of mitosis. The point of mitosis is to create cells that are exactly the same as the cells that came before. All right. Now, all of that is interphase. All of that is interphase. So what do we have left? Well, what we have left right here is this part right here. This is mitosis. Now, mitosis is almost all of this. There's this little slice over here that is this last part we're going to talk about. But mitosis itself is divided up into 4 parts. 1, 2, 3, 4. Mitosis itself is divided up into 4 parts. Now you're going to want 2 colored pencils or 2 different colored pens, or draw them dark or draw them light. Like whatever you got to do, you're going to need that right here. Okay? Everybody got their stuff? All right. So let's do mitosis now. Mitosis happens in 4 phases. Now, the first phase is called prophase. This is the first phase of mitosis. Even though interphase is called a phase, it's not a part of mitosis because the cell is not dividing. Okay. So what happens in prophase? First is that the DNA condenses. Condenses means that it gets smaller. I can't spell condense. It means that it gets smaller. Now what that means is that the DNA gets all wound up around these chromosomes. And so you end up with this shape that looks kind of like this. And what this is it's 2 exact copies of the same chromosome. These 2 things are exactly the same as one another. And if I was to zoom in on this here, what I would see is it would look a lot like this, where the DNA is all wound up around those histone proteins. In fact, the DNA doesn't just get wound up. The DNA winds up, and then the winded up DNA gets winded up around itself, and then the winded up DNA that's winded up around itself gets even more wound up around itself. And then the winded up, wound up parts get wound up around each other. So that whole process of that super winding is called supercoiling. And it becomes so dense and so big that you can actually see it under a microscope. So these are identical pieces of DNA. So this piece on the right is exactly the same as the piece on the left. And because they are exactly the same as one another, they are called sister chromatids. Sister chromatids. This whole thing together is called a chromosome. Now, we haven't even drawn our cell yet. We're going to have to take a step back in just a second and actually draw our cell. Okay, how are we doing here? Is everybody all set? Okay. Just as a piece, just a reminder, what do we call that part in the middle where they join? It's where the bend is, and that's called the centromere. The centromere. All right. So that was just about the DNA. We haven't even talked about like what's going on in the rest of our cell. So let's outline a couple of things before we actually go and draw the whole cell. And let's just outline what's going on in the different phases, okay? So in prophase, the DNA is going to condense down into this chromosome form. That's through a process called supercoiling. All of the sister chromatids, which are exact copies of one another, will pair up at the centromere, and that's called a chromosome. Okay, so after the DNA condenses, the nuclear membrane is going to break apart. The nuclear membrane is going to break apart. And then there's these little things inside of the cell called centrioles that are going to move to the opposite ends or poles of the cell. Opposite end. And they start to form these things called spindle fibers. Now, the spindle fibers are kind of like the fishing line that are going to be used to pull these chromosomes apart from one another. The chromosomes being the fish, if these are the fishing lines. That's the hardest phase. That's the hardest phase to remember everything for. Let's talk about the next phase, which is metaphase now. Metaphase. So in metaphase, those spindle fibers will attach to the centromere of each chromosome. And they will kind of push and pull these chromosomes around until the chromosomes line up along the middle or the equator of the cell. All right, that's it for metaphase. Anaphase. The spindle fibers are going to start shortening now. They're going to start breaking down a little bit at a time. And as they shorten, they're going to pull and separate these sister chromatids apart, so that each sister chromatid goes to its own end of the cell. Now what you have is, since each chromosome is a pair that's an exact copy, and if they're moving to opposite ends of the cell, you are making sure that you get one of each chromosome going to each end of the cell. All right, which gets to our last phase of mitosis, which is telophase. Telophase. And in telophase, the spindle fibers are gone. They go away. They dissolve. The chromosomes will decondense. And when they decondense, you can't really see them under the microscope anymore. They uncoil. They unwind. The nuclear membrane reforms. And then our last process starts. Now, all of this is mitosis. All of this right here is mitosis. What we haven't even gotten to yet is how the actual cell splits apart. The actual cell. What do you mean the actual cell? Well, all we've done is move the DNA to the different ends of the cell. We haven't actually split the cell yet. Mitosis is just the dividing of the nucleus. which brings us, if we go back up to our picture here, you remember how I shaded in this red part right here? I said this is this last thing. So this last part here is called cytokinesis. Cyto meaning cell, and kinesis meaning, like, think about kinetic energy or moving. And that's this last part that happens, which is called cytokinesis. So mitosis is when the nucleus splits apart, but cytokinesis is when the actual cell or cytoplasm is divided, and the cell splits into two identical, what we call daughter cells. So this is when the cytoplasm divides, and this forms two identical daughter cells. Two identical daughter cells. So this kind of starts at the end of anaphase, really picks up into telophase, and I don't know why I paused there. And in animal cells, it just kind of involves like the cell going from this to kind of stretching in the middle, and then to finally, to pinching off into two smaller cells. Now, this is in animal cells. In plant cells, it's a little bit different. In plant cells, what happens is that the plant cell just literally builds a wall in between the two cells. So here's the cell, and then you start to see this wall forming right down here, and then finally you end up with two separate cells, because a plant cell, which is surrounded by that hard cell wall, can't really divide that easily. Okay, let's draw this out now. And I figured out how to add pictures into my little things here. So let's add a picture in here. Here it is. Okay, this right here is an actual picture of the different phases of mitosis. So let's review right here. This is interphase. Whoops, what am I doing here? I need to draw. This is interphase. You can't see the individual DNA. The nucleus looks pretty blurry. Those dark spots are called the nucleolus. That's where the ribosomes are made. This is prophase, and you can see that there's some white space in here. I'm going to color it in yellow, but there's some white space right here. And the reason there's that white space is the DNA has like coiled down into its chromosomes. Remember that during metaphase, the chromosomes line up along the middle of the cell and the equator. And in anaphase, these are pulled apart. Finally, we have telophase, where the nucleus reforms. We can't see our stringy chromosomes. And then really right here along the middle, you can start to see that cell wall forming, because this is an onion cell that's happening right here. So how would we draw this? Well, let's start off here. All right, I'm going to erase some of these lines here. Okay, how do we draw this? Let's start off by drawing interphase in G1. So we're going to have our cell, we're going to have our nucleus, and in our nucleus, let's make this have two unique chromosomes. So in our N is going to be 2. This means two unique chromosomes. And we will make these chromosomes a short one and a long one. This is a very, very simple organism. Two unique chromosomes, short one and long one. However, we have to make sure that we draw them in some unique colors, because one set of this DNA came from mom and one set came from dad. So I'm going to be drawing these as black and red. It doesn't matter which one is which, as long as we realize that, like, there's one of each. So this right here, this is our 2N set of chromosomes, because we have two of each. We have two short, two long, and in those two short, one is from mom and one is from dad. So I'm just going to kind of draw these like this here. They don't have to be right next to each other. As long as there's too short and too long in there, one of each color. That's interphase in G1. And then we have interphase in G2. Now remember our picture over here. What has happened in between G1 and G2? The S phase. So we have to show that our DNA has doubled. So we're going to draw our cell. We're going to draw our nucleus. Instead of 2 short and 2 long, we need to have 4 short and 4 long. So we are going to have 2 short black ones. We are going to have 2 short red ones. We need to have 2 long red ones. And we need to have 2 long black ones. At this point, a lot of you are regretting how big you made your cells, and you're going to want to pause the video so that you can erase everything, curse me a couple of times, maybe even more. I'm looking at you, Courtney. And yeah, that's that. All right, so that was interphase in G2. Let's go on now to prophase. So prophase, if we remember what happens over here, our nuclear membrane breaks apart. We are going to have some centrioles that go to opposite sides of the cell, and we're going to have our chromosomes being all paired up. So I'm going to show my nuclear membrane breaking apart by dotting the nucleus right here. We are going to show that we have 2 long chromosomes paired up that are black. Our 2 short chromosomes are going to be paired up. Same here. And then we've got our 2 long ones. Okay, so how do we draw these centrioles? The centrioles are literally like these 2 little Greek columns that are at right angles to each other. Now, we're not going to draw like Greek columns, but what we are going to draw is 2 little right angles, 2 little rectangles that are at a right angle to one another. And they are going to form these things called spindle fibers, which are just like little strings that are coming out of them. Just little strings that are coming out of them. All right. So we have centrioles, and we have spindle fibers. Right? Next part. We go to metaphase. Oh, that should have been written in black. I'm a terrible person. Okay. And in metaphase, our centrioles are still over here in the corners. And our spindle fibers have attached to the centromeres of each of these pairs of chromatids, each of these chromosomes. And they have pushed and pulled and pushed and pulled until they have lined up all of these chromosomes along the middle. Now, the order that they go in is completely unimportant. I did black, red, black, red. If you did black, black, red, red. If you did black, red, red, black. Like, it doesn't matter. It really doesn't matter. The 2 little ones don't have to go together. We could have drawn them big and short. I mean, this is random. As long as they get lined up like this is really okay. So we connect our spindle fibers to the centromeres of our chromosomes here. And these guys will push and pull and prod them until they get to this middle part right here. Now that's metaphase. So now we've got our arrow. Let's move on to anaphase. And in anaphase, our spindle fibers are going to start to shorten, which then separates the sister chromatids from one another. So I've got my centrioles here. And I'm going to shorten these spindle fibers a bit so that these chromosomes can be pulled apart from one another. And the reason I'm drawing them the way that I'm drawing them is because imagine if somebody was to grab your belt loop from behind you and yank you really hard. your waist and your butt would kind of go backwards while your arms and legs kind of go forwards. And you form this kind of V, and that's sort of what I'm drawing here. Now I want you to notice something. As I draw these, do you notice how this side over here is going to get one long black, one short black, one long red, and one short red? And this side here also gets a long and short black and a long and short red. And if we go up here to our parent cell, what do we have? Long red, long black, short red, short black. Oh my God, we're going to create two individual cells that are the same exact as their parents. That's the point. All right, now we get over here. We go to telophase. It's hard to draw our DNA unwinding, so we're going to kind of keep it together. And now we're going to draw our cell kind of starting to separate here and pinch off in the middle. Let's see what happens in telophase. The spindle fibers are going to dissolve. The chromosomes are going to decondense, and the nuclear membrane reforms. So we're going to draw our nuclear membrane starting to form again. We still have a centriole over here and a centriole over here, but there's no spindle fibers. And, I mean, take a look at our picture. We should have a long black, a long red, a short black, and a short red over here. Long black, short red, long red, short red, short black, long black. And finally, our last part, our cell itself, the cytoplasm is going to divide. This is cytokinesis. We now are going to have two daughter cells that should be exactly the same as what we started with up here in interphase G1. They each have their own nucleus in them with a long and short black chromosome and a long and short red chromosome. And pow, we just drew mitosis happening. Awesome. So a lot of notes for this one, And this is all just to make sure that we understand mitosis. The next thing that we're going to do this week is understand what happens when mitosis makes mistakes that leads to cancer, and how scientists can take a look at just like the pictures of some chromosomes inside of cells and make a decision about whether or not this particular cell might, or this particular bunch of cells might be in a cancerous state. Okay, that is it for today. Everybody be safe. Everybody pay attention to the reminds, and have a great day.